So we're now on to the last lecture for the nucleic acid lesson. So this is the end for the coverage of your second exam. So nucleic acid biotechnology techniques is largely employing the different techniques so that we can study or isolate nucleic acids, or particularly to characterize them. So one of the ways with which we can characterize them is through their size. So in this case, we are using agarose gel electrophoresis. So agarose gel electrophoresis works the same way as your polyacrylamide gel electrophoresis in proteins, except that in this case, you are using agarose as your polymer. So in the same way, you have here your molecular weight ladder, and this is your sample. So we are isolating the fragments based on their size. So your DNA, due to the phosphate groups, are largely negatively charged. So because they are negatively charged, they are usually traveling towards the positive electrode. And they are separated based on their size, such that if this is your molecular weight ladder, if this is, let's say, 100 base pairs long, and let's say this is 50, and then somewhere here is 40. So if this fragment aligns with this fragment, then we can say it is about 40 base pairs long. And similar to your page, because of the polymers, it creates pores such that if you have a longer DNA, it's harder for it to go through the pores. So which means that the migration of our DNA is from larger to smaller sizes, meaning the smaller sized fragments travel faster compared to the larger sized fragments. So this is another representation of an agarose gel. So if you will see, here are the different migrations of the ladders. And you can also say that these fragments here are pure samples because they only have a single band, whereas these can have about 3 or so fragments. And I think this is the molecular weight ladder. So this is a video on how agarose gel electrophoresis is run. So this is your gel, and see they're loading the samples in. And now they are viewing the samples. Oh, sorry. There, and then it will give you this result. So the next technique we're going to study is polymerase chain reaction. So polymerase chain reaction is basically replication, DNA replication, but inside a tube. Which means that for you to do DNA replication outside of the cell, you will need a DNA template. You will need a primer already synthesized for you. And in this case, this primer can be made out of DNA already because you are already making this for yourself. You will need DNTPs as the precursors for your template. You also need magnesium, a source of magnesium, because remember that your polymerase needs magnesium to operate. You need a buffer to mimic the cellular environment, and you also need your DNA polymerase. Now, usually this polymerase came from TAC, or Thermus aquaticus, which is a thermophile, meaning it is an organism that lives in really hot conditions, largely because in polymerase chain reaction, we are cycling through 3 different temperatures, which are largely at around 95 degrees Celsius, 72, and something that's a bit lower than 72 degrees Celsius. Now, the reason why we have these really extreme temperatures is to help with the unwinding of the DNA. So instead of using a gyrase or a helicase, we just use the temperature to separate the DNA strands. So that's via melting. So that's why we need a heat-stable DNA polymerase. So let's go through the video so that you can better see how polymerase works.
The polymerase chain reaction, or PCR, amplifies a specific DNA fragment from a complex mixture. First, the mixture is heated to separate the DNA strands. 2 different specific oligonucleotide primers are added that are complementary to short sequence stretches on either side of the desired fragment.
So if you will see the 95 degrees, it separates the fragments. So we also call this DNA denaturation or DNA melting. So when you say melting or denaturation, the strands separate from each other. So if you will remember, AT will have 2 hydrogen bonds and GC will have 3 hydrogen bonds. So meaning it will be harder to separate DNA fragments that have more GC bonds compared to the AT bonds. So now at 95 degrees, or sometimes some would even use 100 degrees, you have separated the 2 DNA strands. Now usually with PCR, you will have a target sequence. And you are able to target them because of the primers that you designed. So if you will remember from replication, primers are needed to start replication. So it's also the same with PCR. So you have the primers here, and we will let them anneal or bind to our DNA target.
After lowering the temperature, the primers hybridize to the DNA, where they bind specifically to the ends of the desired target sequence. A heat-stable DNA polymerase and nucleotide triphosphates are added. The polymerase extends the primers and synthesizes new complementary DNA strands. At the end of this first cycle, 2 double-stranded DNA molecules are produced that contain the target sequence. This cycle of events is repeated. The mixture is again heated to melt double-stranded DNA. The primers are hybridized, and the DNA polymerase synthesizes new complementary strands. At the end of the second cycle, 4 double-stranded DNA molecules are produced that contain the target sequence. In the third cycle, the mixture is heated, the primers are hybridized, and DNA polymerase synthesizes new complementary strands. At the end of the third cycle, 8 double-stranded DNA molecules are produced that contain the target sequence. 2 of these molecules are precisely the length of the target sequence. In future cycles, this population increases exponentially. Cycle 4. Heating, hybridization, DNA synthesis.
Okay, so I would like to emphasize that this is the target sequence, which means this is the product that we really want. And these longer products are rejects, basically. But with the continued cycle, this will get amplified, meaning we get more copies of this fragment. So the reason why this is called a polymerase chain reaction is because this is a chain reaction. It's a cycle. You repeat these in several cycles and it utilizes a DNA polymerase. So if we continue on with the fifth cycle and so on.
At the end of the fifth cycle, there are 22 double-stranded DNA fragments of the correct length and 10 longer ones. Cycle 6, 10, 15, 20. After 30 cycles, there are over 1 billion fragments of the correct length, but only 60 longer ones. The product, therefore, consists of essentially pure target sequence.
So which basically means that at the end of the 30 cycles, we only get the population that we get the most is that of the correct fragment. That's why all the longer fragments or the incorrect ones, we don't mind them so much because they're not detected anymore. So that's why they're saying it's an essentially pure mixture. So if you will find with each DNA replication or DNA cycle, the DNA fragment is multiplied by 2, which means that we can calculate the number of DNA fragments we get at the end of a polymerase chain reaction based on the equation 2 raised to n being the number of cycles. So let's say, for example, you have already 10 correct fragments of DNA. So meaning the template DNA is already the correct fragment, this fragment already. So if you want to amplify that in, let's say, 25 cycles, then that means at the end of 25 cycles, we have a total of 10 times 2 raised to the 25th copies of that DNA. Because we initially had 10 copies, and each of these 10 copies will be multiplied twice with each cycle. So that's times 2, times 2, times 2, times 2, and so on and so forth, raised to the 25th. Now, because that's already the correct fragment as the template. So this is how we set up a polymerase chain reaction.
You can do PCR in reaction volumes of 10 to 200 microliters in small reaction tubes or microplates. We recommend using thin-walled reaction tubes or plates designed for PCR. There are 7 components in a typical PCR reaction. If you are doing multiple PCR reactions to minimize repetitive pipetting errors and cross-contamination, we recommend making a master mix that includes all of these components except the template DNA. For a single 50 microliter PCR reaction, you will need to add the following volumes.
Okay, you don't have to memorize the final concentration or the volumes, okay? This video is just to show you how we prepare a PCR reaction.
Of each component to a sterile 0.5 ml PCR tube on ice. Place the primers, DNTPs, template DNA, and polymerase on ice to prevent degradation. To assemble the PCR reaction, it is best to start with the component that is highest in volume, such as water, and end with the one lowest in volume, like the enzyme, as it is easier and more accurate to add very small volumes to liquid in a tube than to an empty tube.
So if you will find this motion will seem repetitive because you can't really see the volumes that you are working with because they're really, really small, like 25 microliters or sometimes 10 microliters or 1 microliter. That's why it's really small. That's why you need these micro pipettes to to transfer the liquid needed, the amount needed into the tube where you're going to do the reaction. So we're going to skip through that part because that will just be really repetitive. She's just going to do it again and again. So this is the tube where you're going to place all your reagents, and these are the reagents basically. And then this is the PCR machine, or your thermocycler, which already has the program for the different temperatures needed for the PCR reaction.
D for thermal cycling.
So there you go. Okay. So next we have RE digestion and ligation. So RE means restriction enzyme. Others would call it a restriction endonuclease because they cut from the inside of the DNA. If this is your DNA, they cut from here or here. Now, the restriction enzymes, they are very specific in that they will recognize a specific sequence. So, let's say, for example, they recognize something like ATT, AATTGC, for example. So, wait, that's wrong. Let's try again. Let's say GAATTC, there. So, they usually recognize a palindromic sequence. So a palindromic sequence will be something that you can read in both strands in the same way. So for example, you have GAATTC. In the complementary strand, it's also GAATTC. So this is usually the type of sequences that restriction enzymes recognize, such that if they cut at this side, let's say, they will cut this fragment this way, so that you generate one fragment that will have a G here and CTTAA, whereas the other fragment will have AATTC and AG here. So something like that. So restriction enzymes have been very revolutionary in a sense that it allows you to isolate specific fragments that you want, so you can cut them out, whereas ligation, which makes use of a DNA ligase, enables you to patch them up together. So in a sense, you're kind of like cutting and pasting DNA fragments. So in this example, you have a DNA fragment that was digested using restriction enzyme. So it generated all these different fragments. And when this fragment was added in with ligase, you see this one intact fragment here because the ligase was able to join them up all together. And then once again, when they tried to cut it again with another restriction enzyme, so if you will find, you've generated the same fragments again. So because of restriction enzyme digestion and ligation, recombinant DNA technology is possible. So if you usually say recombinant, that means that you have a DNA fragment from another organism that was added into a different organism. So sometimes people would say the term transgene. Something is only transgenic if the source of the DNA came from a different species. So for example, in BT corn, the DNA fragment came from BT, which is a I think it's a bacillus. It's a bacteria, and so this DNA fragment was cut out of the genome of the bacteria, and placed inside corn. So if that's the case, this is a transgene because this gene, Bt, came from a bacteria. It did not come from corn. It came from something else. So we call this a transgene. So Bt corn is called a transgenic plant or a transgenic organism. So here is how recombinant DNA technology can be made.
First step in cloning a gene is to isolate the DNA from the organism that contains the desired gene. The isolated DNA is purified and then fragmented with a restriction enzyme. Restriction enzymes used in cloning produce staggered cuts in specific sequences in the DNA, generating fragments with cohesive ends.
So when you say cohesive, it's basically that the ends have overhangs which can bind with another complementary DNA.
Each fragment has a single-stranded sequence of nucleotides on its ends that is capable of hybridizing with DNA that has been fragmented with.
So here, if you can see this is a cohesive, or also known as a sticky end, because it has GATC here as an overhang, which means that if you have a DNA fragment with an end that has a CTAG, it can hybridize or connect or hydrogen bond with these ends. And this one as well, if you have a GATC, it's going to bind there as well.
The same restriction enzyme. The DNA fragments are then incorporated into plasmids. The type of plasmid used for cloning has a single restriction site, and when cleaved by the restriction enzyme, generates the same cohesive ends that are in the fragments of the DNA to be cloned. The cohesive ends of the plasmid and DNA fragments now line up, and the enzyme, DNA.
See? So because these are sticky ends, and these are sticky ends, sticky ends, and sticky ends, and because they are complementary to each other, they are able to hydrogen bond with each other. And now DNA ligase can come in to seal the nicks in the backbone. So it's really like cutting and pasting DNA fragments.
Ligase is used to form phosphodiester bonds. The next step is to incorporate the plasmids into bacterial host cells by transformation. Each cell contains a different segment of DNA from the original organism. Taken together, these cells represent a DNA library. The cells can now be plated out on an agar medium.
So when you say DNA library, it means all of these. So usually these inserted fragments came from the genome of a single organism. And if you want to store a copy of that genome, usually you cut it up into these little fragments and put them in a plasmid. So this is a plasmid. It's a circular DNA that is not the genomic DNA of your bacteria. So usually if a plasmid has if a bacteria has a plasmid, it gives them additional characteristics, or characteristics that let them survive better than other bacteria. But in this case, we take advantage of this plasmid because usually bacteria like to get plasmid from its environment. So all of these collectively are known as a DNA library. If a library is a collection of books, this is a collection of different DNA fragments.
Colony of cells containing the desired cloned gene can then be identified and isolated.
And so Why is recombinant DNA technology important? It's important because we are able to produce proteins that used to be really hard to get. For example, insulin. In the olden days, people who require insulin, people with diabetes who need insulin, the insulin has to be taken from cadavers or from other animals. But because of recombinant DNA technology, we can integrate, for example, this gene, which encodes the B chain, and this other gene, which encodes the A chain, and put them inside a plasmid. So this is a plasmid, and this is also a plasmid. These plasmids can be put inside the bacteria, and then you let the bacteria do the work for you, because the plasmid will contain the promoter region and things required for transcription and translation. And so now you just let the bacteria do its work. Let it transcribe and translate your protein, which eventually you can isolate as in this case, your B chain and your A chain. And when they are mixed together, they form insulin. So this is one of the reasons why insulin is cheaper now compared to before, because now we can mass produce insulin inside bacteria. And usually this bacteria is E. coli. So E. coli are very easy to grow. If you just put them in a nutrient medium, they just grow really well. So they're very easy to make happy. So this is another video on recombinant DNA technology. So you have your plasmid here, you have your DNA of interest here, and then you recombine them. So together, this is a recombinant DNA molecule. So that's a specific DNA. And there's your restriction enzyme. So it cuts up the fragment and sticky ends are now generated. Here is your DNA of interest with the same sticky ends so that they can hybridize together. And here is your DNA ligase, which seals the the sugar phosphate backbone. Now this entire plasmid, or this entire recombinant DNA molecule, will be placed inside bacteria. So if I let this bacteria grow forever, each time it splits, it makes more copies of the DNA. So this is what is called molecular cloning. And so this is, let's say, the medium, or the nutrient medium where you've put your cells. So an expression vector is basically a plasmid that contains the promoter regions or the transcription units, so that transcription can happen. And this is RBS, or the ribosome binding site, which is something we also know as the Shine-Dalgarno sequence. And we have the start codon and the stop codon. So that once you put them inside bacteria, and if you grow bacteria in the culture medium or in the nutrient medium, it will be able to transcribe and translate the DNA of interest so that it can produce the protein of interest, which is this one. There you go. So here is a summary of recombinant DNA technology. So it's very straightforward. You cut your vector, you cut the DNA of interest, you put them all together, and you put them inside your bacteria, and you let the bacteria do the work for you. So other than recombinant DNA technology, we have ways to identify the primary structure of DNA. So one of the ways is via the Maxam-Gilbert method. So in the Maxam-Gilbert method, let's say you have this DNA fragment. You put them in a tube which contains chemicals that cleaves at every A and G. And this one cleaves at every G, and this one cleaves at every C, and this one is at every C and T. Now they usually cleave before. So in this case, if let's say you have a segment that says PGCTACGTA, so if this one cleaves at every A and G, so let's say you get this P, and then you get G, and then C, and then T, and then once it counters A, it cuts here. So you get this fragment, GCT, and then so on and so forth. Now, because you generate differently sized fragments, if you run them through the gel, they will assemble based on the smallest towards the highest molecular fragment. So based on this, we can identify that one of the first bases would be a C, this one here. So if you can see in the C column, it is you generate a fragment. Also in the C plus T column, that's how we know that it's a C. Followed by a T, because there's a fragment here, but there's no fragment here. Followed by A, because the fragment is only found in the A plus G column. Followed by C, and then by G, which is because of this segment here. These two fragments here found both in the A plus G and the G column. And then you have T, and then you have A. So based on this, we are able to get the DNA sequence. So this is from 5' towards the 3' region. So this is for the Maxam-Gilbert method. So if you will find, this is the part where we start reading it, because this is too short to even be detected on your DNA gel. The other method is the Sanger D-deoxy method. So it's called a D-deoxy because both the 2' and the 3' positions do not contain a hydroxyl group. Now, because the 3' region does not have the hydroxyl group, you cannot add any new nucleotides to it. Hence, if it is incorporated into your growing chain, it terminates the sequence. That's why the Sanger D-deoxy method is also called the Sanger chain termination method. So this is usually done in a PCR so that you have the template here, and then you have the primer here, which means that all your subsequent DD-TPs, so D-deoxy TPs, or D-deoxynucleoside triphosphates are incorporated here. Meaning the sequence we get is the sequence that is complementary to the template. So it's a sequence of this strand, not the template strand. Okay? So the result we get is from that of the complementary strand. So let's look at this for example. So you have GT, and then this is supposed to be an A. So in the part where you have a mixture of all the normal D and TPs, you added in a DD-ATP, or a D-deoxyadenosine triphosphate. So which means that whenever a DD-ATP is incorporated, the nucleotide chain terminates. So at every A, the chain terminates in this case. So GTA stop, and then GTA CCA stop. So you will generate something like this. Whereas in the mixture that contains a DDGTP, it will stop at every G. So that's why if you get here GTA CCA G, it stops there, and so on and so forth. So these are the D-deoxy molecules found in each of these tubes. And so they generate all these different fragments, which if we run in the gel, will give you different bands, such that the smallest band would represent the smallest fragment. The fragment at the bottom end will represent the smallest fragment, which in this case is GTA. And because we find this fragment at the A column, that means that is A. Sequence there is A, followed by C, 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 A, G, C, T, G, C, T. Which means that this is a sequence of the complementary strand to the template, and it is read from the 5' to the 3' end in this way. Okay, so now the Sanger method incorporates fluorescing markers. So some of them will incorporate yellow for the DDCTPs, purple for the DDGTPs, blue for the DDATPs, and red for the DDTTPs. So that now it's easier to read them just based on their color. So this here is called a chromatogram. So usually if you have DNA sequenced, the person who analyzed the sequence for you will just send you back this chromatogram, and from there you will be able to derive the sequence that you need. So we're just going to discuss a bit about complementary DNA, or cDNA. So mRNA is a less stable molecule compared to DNA. As such, if you want to do experimental procedures with mRNA, it is usually reverse transcribed. So when you say reverse transcribed, that is from mRNA to DNA. So if you remember, transcription is usually from DNA to mRNA. So if you reverse transcribe mRNA, you are getting its DNA counterpart. And because of that, you are creating the complementary strand, or the complementary DNA to mRNA, and that's why this is called a cDNA. Now, this is a mixture, largely because you have this mRNA and this is the DNA part. So the next thing is that you do a PCR so that the DNA part can have its complementary strand, so that you get this. So this is a cDNA. It's basically a more stable form of the mRNA. So it still has the same sequence as your mRNA, but now it is in the DNA form. That's why it's easier to work with it. So here, this is another method called the restriction fragment length polymorphism. So in RFLP, usually you just take advantage of restriction enzyme digestion, so that if, for example, here you have a DNA with mutation in the gene, so this part here is a mutation. Because with a normal gene, this is a restriction site, so meaning you should be able to cut here so that you can generate two fragments. So when you run them through the gel, you get these two distinct fragments. Whereas, because this part here has a mutation, therefore it is not cleaved, or it is not cut by the restriction enzyme. So you get here one single fragment. So this is restriction fragment length polymorphism, which is also used for identifying the parents of a child. So, for example, here you have parent 1, which was cut by a restriction enzyme, parent 2, and the child. Now, supposedly, if you are the child of both parent 1 and parent 2, you should be obtaining a copy from parent 1 and also a copy from parent 2. So this is an example of restriction fragment length polymorphism, which is usually used for paternity tests. So let's say, for example, this here is for the child. And let's say this is for mom. And this is for dad 1 and dad 2. So if this were the data and this is the child, so it will have similarities with mom 1, and this will have similarities with both dads. So it will be hard to distinguish who the real dad is. However, if you have, let's say this is child, this is, wait, let's try that again. If this is child, this is mom, and this is dad, because you have this similarity and this similarity, you would know that this is the dad for this child and not dad 2 here, which does not have shared similarity with the child, assuming this is how the samples were loaded onto the gel. So there are several omics technologies that you can look into, which we will not discuss further here. I just need you to know that they exist. For example, genomics would be any study that will involve the entire genome of any organism. So if you say it's a genome, it would incorporate everything in that DNA. Proteomics would be all the proteins found, it's a study of all the proteins found inside an organism. Transcriptomics would mean all the RNA found inside an organism, and metabolomics would be the study of all the metabolic chemicals or metabolites basically found inside an organism. So this is where we're going to end our discussion on nucleic acid techniques.